Hey everyone. Welcome to The Whiteboard Biologist. Today, I am going to talk about Pathogenicity Islands, that is also known as Pathogenic Islands. So first, what is a pathogen? A pathogen is an organism that can cause an infection. And the ability of a pathogen to cause infection is called pathogenicity. Now look at the Pathogenicity Islands. Pathogenicity Islands, or PAIs, are a distinct class of genomic islands that are acquired by microorganisms through horizontal gene transfer. This means that they are not inherited from parents, but rather acquired from other microorganisms through mechanisms such as conjugation, transformation, or transduction. Please remember it, PAIs are not plasmids and were first discovered in pathogenic E. coli. PAIs encode various virulence factors, which are a wide range of mechanisms employed by microbes that enhance their ability to establish infection, spread through host tissues, and destroy the host immune system. They have a unique GC sequence, and their size may vary from 10 to 200 kilobasbar. They are flanked by 16 to 20 kilobasbar of direct repeats and insertional sequences, IS, that enable insertion in DNA. They also have an integrase gene and tRNA, which act as sites of recombination into DNA. They are normally absent from non-pathogenic strains of the same or closely related species. Salmonella is known to have at least 5 PAIs, which contribute to its virulence. PAIs carry functional genes for adherence factors, toxins, the iron uptake system, invasion factors, and the secretion system. Now look at some factors encoded by PAIs. Okay, let's talk about the mechanism of PAIs. As mentioned before, PAIs are genomic fragments harboring various virulence genes in pathogenic bacteria. Some PAIs can be excised from the bacterial chromosome, known as excisable PAIs. This ability is due to the presence of directly repeated sequences, or DRS, in their two ends, also named ATTL, or left DRS, and ATTR, or right DRS. DRS or ATT sites are recognized by integrases and excisionases or recombination directionality factors that catalyze the excision. The excision of the PAI generates an episomal element that contains one of the ATT sites, or ATTP, while the other ATT site remains in the chromosome, or ATTB. The basal excision rate is low, but it can increase due to changes in pH, temperature, intracellular environment, and oxidative stress. An increased excision rate could lead to a change in the expression of the island genes, and also increase the passage of the episomal element to other bacteria, through lateral gene transfer, conjugation, transformation, or transduction. Now, let's look at some examples of PAIs. Salmonella has something called SP1 and SP2 sites. Helicobacter pylori have 2 strains, one of which is more virulent than the other due to the presence of the CAG pathogenicity island. Yersinia pestis high pathogenicity island 1 has genes regulating iron uptake and storage. Rhodococcus equivirulens plasmid pathogenicity island encodes virulence factors for proliferation in macrophages. So, That's a brief overview of PAIs and how they work. I hope you found this video informative and helpful. Please don't forget to like and subscribe for more content like this, and I'll see you in the next one.